show please the slides, and then I will announce and you'll start. Yes, so it's a pleasure for me to introduce our today's speaker, Dmitry Nечипуренко from the Moscow State University, with the title of the presentation, Computational Modeling of Hemostatic Response to Various Types of Microvessel Injuries. Please, Dmitry, you can begin your presentation.
Thank you. If you allow me, I will turn the camera off, so it would be easier to present. Okay.
As you like.
Okay. Okay, first of all, thank you for inviting me to this seminar. It's a great pleasure to be here. So I would like to thank, first of all, Vitaly and also Anastasia, who welcomed me and helped me getting here. So today I will speak about our efforts to answer several long-standing questions using in silico model that we recently developed. So my talk will consist of several parts. First, a brief introduction to our lab, to the problems that we're tackling. Then I will talk about some mechanical problems and how we solved it, mechanics of interplatelet interaction and how they explain the dynamics of thrombus that we observe using in vivo models. And in the last part of my talk, I will focus on the last and more complicated model that we use to understand how thrombi forms under different conditions. Okay, so my lab, which is called Biorheology and Biomechanics Lab, is a part of the Institute of Theoretical Problems of Physicochemical Pharmacology. And the main people in this institute currently are Fazliya Tolokhanov and Michael Panteleev. Michael Panteleev, whom probably some of you know. So here you can see the graduate students on top and students below. So generally we have a lot of young people in the lab. Most of them are from Lomonosov Moscow State University, but not all of them. So basically what we're trying to understand is how the hemostatic response occurs under different conditions. For example, we know that in case of non-penetrating injury of the vessel wall, we see that some thrombus forms, and we will speak about the dynamics of such thrombi. If we increase the severity of the injury, for example, if we penetrate the vessel wall completely, there will be the blood loss, and in this case, the hemostatic plugs forms to the side. And now there are models that study also these types of the injuries. And one of the most interesting in terms of hemostasis is the injury when we have complete vessel transection. And in this case, we know that the plug forms and has to completely stop the blood loss from the vessel. And generally, most of time of my talk, I will focus on this type of injuries, which are more associated with thrombosis, because in this case, unrestricted response can lead to vessel occlusion and problems. For example, this may lead to myocardial infarction or ischemic stroke. And so basically these things are probably most well studied because of their clinical significance. And now we will have a look at how people usually study it. For example, using in vivo models in mice. This is a famous paper by Tim Stalker and Lawrence Brass from University of Pennsylvania. Here you can see that upon the laser-induced injury of small vessel in mouse, we can see that big thrombi forms. Here red are platelets and blue is fibrin, so the system work all together. And we see that at some time this thrombus stabilizes and doesn't stop growing. Interestingly, we can see the same dynamics in the bigger vessels. So the former vessel was arterial, so it's around 30 microns in diameter. And here we can see pretty much the same dynamics in a mouse artery. This is the carotid artery of a mice. Here, chemically induced injury leads to pretty much the same dynamics. So first, thrombi grows really big, and then it disassembles. Some ruptures happen, and in the end, we see just a small thrombi here at the side. So basically, currently, we call this a three-stage dynamics of thrombi formation. First stage is growth. Then it diminishes or shrinks in size. It's phase number 2. And phase number 3 is what we call stabilization, when it doesn't change for a pretty long time. So, and of course, one of the big goals here is to understand what happens here and how to explain such three-stage dynamics which happen under normal response. So when no occlusion happens, we see this three-stage dynamics. So basically, if we have a look at books from surgeons, for example, they consider 3 degrees of vessel wall injury. So first one, non-penetrating. So in normal cases, we don't have occlusion here. This is a normal response. The second degree is penetrating, but partial penetration. It's type 2. And in this case, if the normal response means that we plug this injury here, we build a platelet plug here, but we don't occlude the vessel here, but we also stop the blood loss with this plug. And the third degree, which is normally associated with hemostasis and complete cut, in this case, normal response suggests that stable plug will be formed here. So the complete to completely prevent the blood loss in this case. So the big question that a lot of teams all over the world try to answer is how the hemostasis system scales its response depending on the injury type. So how it happens that here, under normal conditions, we don't have a vessel occlusion, but here hemostasis system under normal circumstances builds the occlusive thrombus to prevent the blood loss. So this is a big question. The more specific question that we are now currently trying to answer is the first one. So what are the possible mechanisms explaining this three-stage dynamics responsible for this three-stage dynamics of thrombus formation that we observe in vivo in various models of non-occlusive thrombus formation? It's the first question. And the second question, so what are the key factors that govern formation of the stable plug in this case? So I will be focused mostly on the first question in my talk, but in the end we will also a little bit speak about the second scenario when there is a complete transaction of the vessel. Do you hear me? Is everything okay?
Yes, we hear you. For me, it's everything is okay. Yes.
Okay, great. Just to make sure that no connection problem. And
yeah, well, it's a little bit. Yeah, I know that it's a little bit frustrating speaking to the screen, but we have to deal with it. Yes, it's okay. You can continue, please.
Thank you. Yeah, thank you. Okay, so in my team we use several methods. So first one is in vitro, like microfluidics or hydrogel-based methods. Basically what we do, we make some artificial channels and we perfuse human or mouse blood through the vessel and observe using microscope what is going on here. So how thrombi form and what is the fate of the thrombi? Do they occlude the vessel or not? So this is one of the basic experimental methods that we use. So of course, being physicists, biophysicists, we use some mathematical models. So we compute them using different computers, supercomputers as well. So in this case, we model from the molecular scale. For example, sometimes we model von Willebrand factor dynamics. It's a huge protein in the bloodstream. And also we use continuous models of clots. But today we'll speak about mesoscale modeling, the particle-based model, when we consider each platelet as a separate entity. This, to my mind, is the most interesting type of modeling that we can do to understand what's going on in the microvessels. But however, this type of modeling doesn't suit for huge vessels like arteries or veins. Here, of course, for now we cannot consider each platelet separately. Okay, so the last years we also started working with mouse models, but it's just the beginning, because of course it's the most complicated type of experimental modeling. But hopefully we will get some interesting data using these mouse models as well. So as people which normally were focused on primary hemostasis and platelets, I would like to emphasize that from our point of view, platelet as a cellular unit of primary hemostasis has its own superpowers. So first, it can adhere at high shear, probably most of you know it. And the second, it can really rapidly respond to various stimuli. So to biochemical cues or biomechanical cues. In response to these stimuli, platelet can activate integrins, which allows it to firmly arrest and to aggregate with other platelets. It can also change its shape. It can secrete granules. It can contract like a small tiny muscle. And also it can become a zombie platelet process, which is called necroptosis, which leads actually to cell death. But in this case, the surface of platelet can become procoagulant to help assembling different coagulation cascade complexes here. And so this is also probably one of the important physiological responses of platelet, but it's still under debate. But of course, no question that integrin activation, granule secretion, and probably contraction is something really important from physiological point of view. So the problem that when we try to understand what's going on inside the thrombi is that here we have a lot of processes going on, and we can distinguish several things. For example, the local biochemical environment. So it actually influences the mechanical interactions of platelets, because this environment leads to different functional responses. But these mechanical interactions, they in turn, they also influence functional responses and local biomechanical environment, because when platelets aggregate, the local flow fields change. And so this is a very complicated process when all these things influence each other, and there is a crosstalk between platelets in terms of biochemistry, because, for example, they secrete different granules, in terms of mechanics, because platelets can contract, and they can change shape. And to understand what's going on, of course, it's important to integrate this platelet responsing during thrombus formation. And for now, one of the most important instrument to do this, of course, is in silico modeling, because we can address a lot of questions using experimental setups like we discussed recently. So what we're trying to do is to integrate these responses using some in silico models. So basically the framework of how we do it is like this. So we first analyze some experimental data on single platelet responses or interactions. This can be our data or some published data. So then we incorporate this data into the computational model. Then we perform simulations and try to generate some novel predictions. And after that, if we're lucky, we or someone else can verify these predictions using experiments. So in first part of my talk, the second part of my talk, I will focus on how we try to link the mechanics of interplatelet interactions with overall dynamics of thrombus shell. So the shell is this outer region of thrombus. If we again look at the experimental data, we see that thrombi behaves very in a very complex way. So here, after laser-induced injury, we can see that a lot of platelets aggregate, but still this mass resembles some viscous fluid because it flows and it has high plasticity. And we first try to understand how to explain such a high plasticity of this outer regions of thrombi. So this is mostly mechanical questions. And we hypothesized that actually this plasticity might be important for preventing vessel occlusion because forces from the flow, pressure-driven and viscous drag can move platelets and move this whole viscous mass and prevent vessel occlusion. So we try to answer how does the mechanics of individual platelet-platelet interactions can explain the fluid-like behavior of these external regions of the thrombi. And to do that, we build a really simple model which has two types of interaction. So first, the stochastic interaction. So in this model, platelets considered as disks, and between this disk, a spring can form with some probability. And we calibrated this interaction using data on how platelets roll along the von Willebrand factor surface. So here is an experimental video showing how in vitro platelet roll if we have a lot of von Willebrand factor covered on the surface. And using videos like this, people have published data on how this rolling velocity of single platelets on average, of course, how it depends on the shear rate. So here, after 1,000 inverse second, we see pretty much linear increase of the rolling velocity. So first we calibrated this stochastic spring interaction using this experimental data. And after that, as the second step, we also introduced deterministic interaction between two platelets with some force field, which we modeled using Morse potential. And this interaction, by this interaction, we mean the interaction through integrins. So platelets, after activation, it can activate its surface integrins, which bind fibrinogen. And through this fibrinogen bridges, two platelets can stably interact with each other. And there are also some nice experimental curves obtained using atomic force microscopy, which show how this force depends on the distance between platelets, and we see that there is some maximum force. So we try to describe this experimental using Morse potential. And so in this model, the second type of interaction with force field actually described the interaction using with activated integrins. So generally, this model, it looked in this way. So first of all, we have some 2D computational domain, and here we have some unicellular layer of platelets which irreversibly attached here. So, and we also have platelets in the flow. So then cells are brought from the flow here, and they can interact with this first layer using this stochastic interaction, which we described by the data of platelet rolling. And also in this simple model, the blood flow was constant, so platelets didn't influence the blood flow, so it was considered constant. And the platelets which were stationary in respect to the blood flow, they experienced the Stokes force, which is suitable for spherical particles, but here we use it to describe the dynamics in 2D. And also we consider that the flow is constant, has parabolic profile. And what is really important is that in this model, we assume that platelet activates with time spent here in thrombus. So this is a really simple model. And this extent of platelet activation, it influences both probability of stochastic spring emergence and the magnitude of this deterministic force by which we describe this interaction through activated integrins. With this, using this model, we try to describe the situation when there is no potent activator like thrombin here, and you see that in this case we see cycles when thrombi grows, then it disassembles, then it grows again. And this model, because here activation just depends on time spent in thrombus, we have a lot of cycles of growth and disruption which just never stop. Yes, because any platelet which gets to the injury site, it will activate regardless its history. So, and if we have a look at the case when we block thrombin activity using hirudin in vivo, we can see that some really the dynamics is pretty much the same as here. So generally we have several cycles of growth, disruption, and then all of a sudden everything stops here, but not in this very, very simple model where platelet activation just depends on time spent in the thrombus. So with using this mechanical model, we showed that in case we consider this stochastic component during the disruption, we see that small pieces of thrombi, they experience some plastic deformation, some shear deformation, like you can see here. But in case if no stochastic component is considered, the thrombi behave differently, so it's just like more rigid pieces which just rotate, and no inside these pieces the distances between different platelets remain pretty much the same. So that means that it's the stochastic component which is important for describing this fluidity of the external regions of the shell. So with this mechanical model, we had several conclusions. So first of all, we understood that this plasticity or fluidity of this thrombus shell of this external regions of thrombi is likely driven by the stochastic nature of primary platelet-platelet interaction. And these results actually suggest that this interaction involves just very low numbers of receptor ligand bonds, because otherwise it wouldn't look like a really stochastic process. And another conclusion is that using the model, we could, and comparing to the experimental data, experimental dynamics, we estimated that the interplatelet forces in this thrombus shell are around nanonewton. So now we know that highly activated platelets experience forces around 10 and 20 nanonewtons. So this probably refers to the internal regions of thrombi. And so interesting that our estimations for external regions are within this lows 1 or even less than nanonewton range of interplatelet forces. So the next part of my talk will be focused on more complex processes involved in the formation of the thrombus, or of the thrombus which is heterogeneous. We know from these experiments that I showed you that actually this thrombi shows high heterogeneity. So there is internal region which is called the core, where platelets are densely packed. When they secrete granules, that means that they are irreversibly activated. And there is also the external region, which is called shell, where platelets are not that highly activated, not that densely packed, and experiments with different inhibitors in vivo show that this internal region in microvessels depends on thrombin activity, while these external regions are dependent on some agonists like ADP or thromboxane A2, which are secreted or synthesized in this internal region, which is called core. So it looks like this core feeds the shell with some fuel which originates within this core, and this fuel is some low molecular weight agonists like NDP and thromboxane. However, we don't understand how to explain this three-stage dynamics in this case. So why does this core stop growing at some point of time, and why this shell first forms really big and then disassembles? So it's not clear, and this is what we're trying to understand. Another important question is what is the function of this internal shell? Why do we need them, this shell at all? It's not clear as well, because using some pharmaceuticals we can get readout of this shell, but still the core forms and polymerization of fibrin occurs within this core. So it's not really clear why at all do we need this huge external shell of thrombi, which at some point disassembles. So to try to understand what is going on, we decided to build some model where we considered two platelet agonists explicitly. It's thrombin and ADP. And among the platelet responses, we considered only activation of integrins, which means that the parameters of interactions between platelets change, and also the secretion of granules. And these granules, they contain ADP. So using this model, we then try to understand, so what is the interplay between the secretion of granules here, the activity of thrombin, which induces this secretion, and also the overall dynamics of the thrombus. So this is the last big part of my talk. So basically, the model is pretty much the same, but it has some important refinements. So in this model, instead of considering that platelet is activated with time, we considered that its activation depends on local concentration of some chemical species. So first of all, it's thrombin, and then it's ADP. So we consider the constant flux of thrombin from the side wall, and this thrombin induces secretion of platelets, and these platelets secrete dense granules, and we consider it as secreted ADP, which also influences platelets in terms of their mechanical interactions. Now I will talk in more details. So basically the first part is pretty much the same as this mechanical model that I described before. So we still have two types of interaction, GP1B1 Willebrand factor interaction described using the stochastic springs. But in this case, we, as thrombi can grow really big in this case, of course, we have to model the change of the velocity field. So to do that, we solve numerically Navier-Stokes equations. So we consider stationary flow, and we recalculate it in case if thrombus geometry changes. And here you can see the example of this solution, of the stationary solution of Navier-Stokes. So to do that, we use open form. and we use Gmesh to build this mesh. So you can see that in this case, we accurately reproduce the intra-thrombus flows in 2D. So the mesh is rather detailed inside the thrombi, but for a given geometry, we calculate hydrodynamics once, and then we calculate the forces which act on each platelet, and then calculate the dynamics of platelets. And if some platelet changes its position, we recalculate the flow. So this is the idea for the hydrodynamics model. The second part, which also changed, is that in order to consider chemical species in this model, we solve Langevin equations for virtual particles. So we considered a lot of virtual particles that can, for example, thrombin, which emanates here in the injury site. So we solved the Langevin equation, and then to get the concentration map, we count how many particles are in different regions of the of the square map, which is 1 micron by 1 micron. So this is done using the CUDA architecture. So we use GPU because it's computationally intense, but we can parallelly solve these equations for many particles, and then have some map of the concentration, and use this concentration map to determine how platelets are change their mechanical parameters and secrete granules. So this, for example, this thrombin map, which is shown here, we see that platelets become activated, for example, by thrombin, and in addition to the stochastic bonds, we see that we also have this deterministic interactions with Morse potential, and this parameter of platelets activation, they are determined by local concentrations of thrombin. And the last and very important part in this model is secretion of dense granules by platelets. So these granules contain ADP, ATP, but we consider only ADP because we know how it impacts mechanical properties of thrombi. So we used experimental data on platelet secretion in suspension of platelets, so where we have a lot of platelets and we add some thrombin, and we see how the number of dense granules change with time. So we show that it can be nicely fitted using exponential curves. And that means that each granule inside platelets has some constant probability to be secreted, and this probability just depends on local thrombin concentration, and we used it in this model. So this secreted ADP is also considered using this virtual particles framework. So this is example of how we see the ADP map in after dense granules had been secreted here, here, and here. So these three secretion events, and we have a snapshot showing the concentration of ADP in here. So this is a rather computationally intense model. It's 2D, but still we have to calculate hydrodynamics often. We have to calculate these virtual particles to understand the concentration maps. So to solve this all these equations, we used Lomonosov 2 supercomputer of Lomonosov, Moscow State University. And now I will get to the results. So what we studied and what we obtained. So first of all, I will focus on this thrombosis scenario when we have some injury site here. And later on, I will show you the results when we model transection of vessels. So in this case, we model that there was a healthy vessel, but all of a sudden there is some cut and the blood starts to flow from this vessel to the outer wall. So here we consider that a platelet plug can form to stop the bleeding. And in this case, these injury sites here and here are the sources of thrombin, which generates and which can activate platelets. So first of all, I will show you the typical dynamics that we see in this model here. Here is the time. So basically what we see that there is a core of platelets activated with thrombin, these blue platelets, and because of secretion there is some ADP which is responsible for the formation of this outer region, which is yellow. Here platelets are activated with ADP, and we see the cycles of growth and the disruption of mostly this outer layer, but also some platelets which are blue in color. So these cycles, they repeat many times. And of course, we didn't really like this dynamics because it doesn't look like what we observe in vivo. But what is interesting is that, for example, if we change the shear rate from 1000 inverse seconds to like 100 inverse seconds, we see that in this case, we see the formation of occlusive thrombi. So the height increases, and all of a sudden the stable occlusive thrombi forms, and the velocity in of the blood in the vessel drops significantly. But in case if we have rather high shear rates like 1000 inverse seconds or 2000 inverse seconds, which are normally considered in arterioles like this, we see these cycles of growth, disruptions, and growth again. So in order to test the idea, so we decided that this happens because thrombin actually in our model is not confined in the direction of the flow. So we see that it's convected downstream, and here blue platelets also originate. So with time we see that the core grows in length because thrombin particles are advected, are brought by the flow downstream. So we decided to see what will happen if we have, for example, spatial inhibition of thrombin in some area downstream. For example, in this case, here is the injury site, and after this coordinate, we strictly enforce that activity of thrombin is set to 0. So this was the first type of modeling. Another was when we perfectly inhibited thrombin, which interacts with the surface. So if thrombin particle gets to the surface, it disappears. So with this, we modeled something like interaction of thrombin with thrombomodeling on the endothelial surface, or interaction with heparin sulfates, which are also on the surface. So nevertheless, so we observed in the experiment that thrombus core is really confined, and we decided to introduce it strictly into the model with these rules. So in this case, what we see is that the dynamics changes. So here we also see the formation of a core, then a shell forms once it disrupts. And after a second disruption, we see that actually no more shell is forming. And if we look at the dynamics of ADP in this case, we see that first the secretion starts, and because of the secretion, this shell grows, but then actually secretion stops, and this happens because the dense granules here are mostly depleted, because all the dense granule in the region where thrombin activity is high, all the dense granules are depleted, and that is why in this case we end up with some stable core and no shell. So to see if we can also look at the plot. So the blue line corresponds to the spatial inhibition of thrombin in case when there is secretion. We see that there is one cycle, the second cycle, and then thrombus disassembles, and we see some stationary level of thrombus height. In case when there is no secretion, we see just a monotonic increase in height, but it stops in this case. And in case if we have no inhibition of thrombi, we see the cycle. So it grows, disrupts, grows again, disrupts, and starts growing again. So with this, we concluded that actually the coordination of this localized thrombin activity and the secretion of dense granules in this internal region of thrombus called core leads to actually to the to stage number 2 and to stage number 3 of thrombus formation. So we see that shell disassembles, and then this core which is left, it is stabilized because here all the granules are already secreted, so there is no fuel to feed these external regions, which can be here only if ADP is secreted inside this core. So we also started to model the case when thrombin is not inhibited in the volume, but it's also only inhibited if it binds here with the surface. But according to preliminary results, the dynamics is pretty much the same. So it means that we can observe the three-stage dynamics even in this case. So first it grows, then disrupts, and then it doesn't grow again. But of course, we need to calculate more to be really sure about these conclusions. And next, after this, we decided to model what happens in case if we have the complete cut of the blood vessel. So here we can see that, so the blood flow is from here, as the arrow shows, and here we see the formation of these platelet plugs. But for now, they didn't occlude the vessel, as hopefully they would at some time. But at 35 seconds that we computed for now, unfortunately, we don't see any occlusion here, though we can see that the vessel lumen here is becoming less. Yes, so here, if we compare to situation when there is no secretion of dense granules, when there is only activation of platelets with thrombin, we can see that here just this platelet, this core forms, which is parallel to the flow, but these cells cannot grow to each other, so to become closer to each other, to eventually close this opening. So with this we concluded that actually it's ADP-driven formation of these outer layers. It's critical to build up the plaque completely here to prevent the blood loss, but still, unfortunately for now, we didn't observe it. This is rather computationally intense problem, and now we're trying to optimize our algorithms so it could be more rapidly calculated, so that we don't have to wait like for a week on the supercomputer to compute dozens of seconds. So with this, I would like to conclude. So first of all, our results suggest that actually the dynamics of secretion might be really important, and we probably can explain this three-stage dynamics of thrombus formation by the fact that thrombin activity is confined within the thrombus, and that means that at some point all the granules will be secreted and the shell will disassemble, and this is what our model demonstrates. And for this, we have a prediction that if we look at the core, we should have all the granules here secreted. So this is the prediction of the model. So and another prediction is that actually this secretion is important to narrow the transected vessel. And actually, we believe that the function of this shell, which is in this case just disassembles, is to close the vessel after the complete transection. So we think that it's really important in hemostasis, but in thrombosis, of course, it can also lead to the vessel occlusion, but normally it shouldn't. Okay, so the future directions of this work. First of all, of course, we hope to analyze this hemostatic clog formation at the later stages. That's what we're doing right now. We also want to investigate the effects of platelet contraction on overall dynamics of thrombus, because for now the thrombus is rather permeable for the flow, so the flow is rather high, and we don't model the contraction. We also want to analyze the effects of thrombin binding to platelet surface, because it might be the possible mechanism of the spatial confinement of thrombin. So for now, we don't understand what is the reason of this spatial confinement. We just had to introduce it somehow into the model, in the direction of the flow. So another thing is that we can study also some other agonists like thromboxane A2. We also want to couple this model with a more accurate model of platelet signaling, which is called virtual platelet. It is also developed in our institute by people from another group. We also want to have a reduced model of coagulation cascade and introduce it inside this model, because for now we have just a constant flux of thrombin, which is of course an oversimplification. So we need more accurate description of the coagulation which is going on here. And in the end, because we have a grant on thromboinflammation, we also plan to investigate how the secretion of interleukin-1 beta by platelets can affect different aspects of thrombus formation. This is also what we plan to do. With this, I want to thank my colleagues. So first of all, Anastasia Masaltseva, who was the main engine for this sophisticated model that I told you in the end. This is Anastasia. I would also thank Valeria Kaneva. Her work was in the beginning of my talk on the mechanics. Peter Trifonov, and of course Fazli Atolakhanov and Michael Panteleev, who initiated this kind of research in my group, and also the Russian Science Foundation for financial supports. And of course, thank you for your attention.
Thank you very much. There was some echo. Well, I try again. Sorry, there is some echo. I don't understand why it is like that. Thank you very much for this really very interesting presentation. And of course, we have many questions to discuss. So please, if there are questions in the audience, we can discuss them. Otherwise, waiting for the questions from the audience, I can already start with some of them. In particular, you mentioned in the first part of your presentation that stochasticity of platelet interconnection, it somehow can influence the fluidity of the thrombus. Could you please comment on that? How we can understand in a simple physical terms how the stochasticity increases the fluidity of the thrombus?
Okay, so how I understand this concept. So okay, for example, if we consider here the simple experiment, do you see the screen now?
Yes, we see the screen.
Okay, so here there is a von Willebrand factor on the surface, and we see that platelets can translocate, and there is some average velocity. But if we take a look what happens in detail, platelets, how they behave. They stuck for some time, and then they go. Then they stuck again. Then they go. So there is some stop time and like go time in this case. And people normally to describe such rolling, they consider rather simple models, like there is some probability of bond creation, and there is some probability that this bond will break up. And the idea is that it seems that this stochasticity is actually what leads to this average velocity, and it's how it depends on the shear rate in case if we have the platelet rolling. If you try to describe this process, for example, with force fields, that there is some potential and platelets interacts with potential. In this case, you won't be able to describe this curve, because in this case, if the fluid force is more than the critical force of interaction with this force field, then the platelet will move really fast. If it's if the force is lower, then the critical force is higher than the flow mediated force, then the platelet will just stuck and stay for forever. So people currently think that it is this on and off type of bonding which is responsible for this type of movement. So, and we try to think that to test whether this type of stochasticity is what drives, what actually drives this fluidity of the platelet aggregate, because we know that pretty much the same types of interactions happens between platelets in the platelet aggregate, and we tried to test it using different approaches. For example, here we considered no stochastic component and tried to have the parameters so that the thrombi size will be pretty much the same. And in this case, we see that thrombi behave differently. They disassemble rather fast. And during this disassembly, if we have no stochastic component, we see that like internal parts, they behave like some rigid body. But if we consider the stochastic component like shown on top, we see that, first of all, that the process of this disassembly is rather slow. You can see it here. So you can see that there is some movement. So it's shuk shuk shuk. It's like it behaves more like a liquid in this case because of this internal stochastic rearrangements. So with that, we decided that it's important to consider the stochastic nature of this primary interaction to explain this plasticity.
Yes, I think it's a very good explanation. I would maybe formulate it just to summarize it. So if there is no stochasticity, then all platelets in some sense behave in the same way. Either everybody goes or everybody stops, right?
Pretty much like this, yeah.
Yes, and because of that, we have more rigid behavior of the whole.
Yeah.
I think it's a really very interesting situation. I know not so many of them where stochasticity gives really something new compared with deterministic models. It's not just some perturbation which on average gives the same, but maybe it's really something different. Please, Anastasia has a question. Please, Anastasia.
Okay.
Yes, thank you. I have several small questions. The first one, you talk about the micro vessels, and my question is what principally changes in the levels? Why do you specify that these dynamics from the growth are in the micro vessels? And the second question, that you have quite complicated models. Is it possible to have some analytics for them? And in any case, how do you verify your models, your results, not physiology, but mathematically? If I understood.
So yeah, I'm not sure that I understood all the questions. So the first question, we model situation in microvessels, and the question is how do we think things are different in bigger vessels? So what is the difference? Is that a question?
Yes, yes.
So basically, to my mind, things are pretty much scalable, and there is no some principle thing that qualitatively changes when we go to the microvessel. I believe, of course, there are blood pulsation which are higher in arteries, that's for sure. But if we compare arteries with arterioles, and you can see it, for example, with this video, yeah. So here we see the formation of this. Sorry, okay. It really matter which video to see. So here the scale is like here 30 microns. We see that first big thrombi grows, and then it becomes smaller, and pretty much the same, to my mind, happens here in artery, where the scale is about 10 times or maybe even 20 times bigger. So in the end, we see just a small core. I think we can also call it a core in this case. So to my mind, things are pretty much similar here. But this is the idea. Maybe I'm wrong.
Thank you. And second question was about if you find some analytical solutions or something like this in such model, or if only the numerical results are possible, and if the last is true, then how do you verify the results and your implementation of your numerical models?
I'm really sorry, but I'm afraid that I didn't understand everything, because your voice is not clearly.
Yeah, Dmitry, I can repeat the question. Do you have some analytical methods to study these problems?
Okay, so there were several analytical methods in the literature, and one was even published in Nature. But currently, if we consider this type of detail that we consider in this model, I'm not sure that we can analytically trace these systems of such complexity. But still, in the literature, you can find some analytical models of thrombus formation, but I think that they are not very suitable to describe behavior like we observe here in the experiments. I mean, is that response clear?
Yes, thank you.
Mm-hmm. Yes, please, Anas, you have a question. Please, you can ask.
Yes, thank you very much for this interesting talk, and I have two questions. So the first one is, I think for the results of the first model, you showed that the growth patterns dynamics for the shear rates equal to 1000 and 2000 are pretty much the same. So I would like to understand this more, because as far as I understand, the force between platelets do not depend on blood flow. So how can we understand this better? So do you have any explanations? I think it's slide 14. Yeah.
This one?
Yes.
So do you mean that, just to make sure, you asked what is my interpretation that for 1000 and 2000 inverse seconds, the dynamics is pretty much the same, or
yeah. Because I think the drag force will change, so I think this will lead to more detachment of platelets. But yet we can see that the two curves are not that big difference. So do you consider like some additional force that depend on flow, which makes platelets more adhesive, or is it because flow is bringing more platelets to the injury sites?
No. So in this case, what we generally see that so it qualitatively it's indeed really close, but quantitatively, first of all, you see that disruptions at 2000 inverse seconds happens at the heights which are systematically lower than the heights for 1000 inverse seconds due to the increased drag force. And another thing that because of this, we see that the formation of like the second cycle, it happened earlier at 2000 inverse seconds. Regarding the speed of growth, it's indeed pretty much the same here and here. Why is that? I'm not sure. So we didn't study it in detail, but the speed of growth, I think here there is some kind of compensation. So first of all, you're right that the flux of platelets is increased, but also the activation of platelets by species is decreased because the concentration experience the flow which is increased, and probably lower concentration of species results in that the speed growth is pretty much the same. But of course, these are just my speculations. I think it should be studied in more detail here. Thank you for this question.
Yeah, I think it makes sense. And can I ask one additional question? Sure. Yeah, so I think in slide 15, I think it's 2 slides after that. So you have shown that, yeah, I think it's here. So for ADP, the production of ADP stops after some time. So maybe I didn't understand the model very well, but why does it stop? So do you assume that the granular production stops after some time of activation, or does it depend on the level of yeah, so how do you interpret this?
Thank you so much. Yeah, this is in the heart of our idea. So platelet has on average around 7 to probably 10 or maybe even 15 dense granules. So if platelet is here in the region when there is a lot of thrombin, it will secrete all its granules for pretty fast. So that is why we see in the beginning that there is a lot of secretion here. You see that a lot of secretion and a lot of ADP. But after, but this number of granules inside each platelets, of course, is limited. It's just like around 5 to 10 granules. And that means that after all these granules are secreted in the region where thrombin activity is high, that means that secretion process overall will drop. Just because this pool, the granule pool, is limited in this core region, and we believe that this is that might explain why we have this disassembly of external region.
Yes, that makes sense. Thank you for the explanation.
No, but to complete this question, maybe I did not understand very well. The limitation to thrombin or inhibition of thrombin limits also this granule production, right? This is what you assume.
Yes. So we think that okay, here the flux of thrombin is constant. It's constant all over the time that we modeled. So here from this yellow region, and here the confinement of thrombin activity with height is due to the flow inside thrombus. So that is why in these outer regions of platelet platelet aggregate, we don't have much secretion because the concentration of thrombin is rather low because of advection. And actually ADP can get here because it has much higher diffusion coefficient. So that is why it's not that highly diluted as thrombin. And here we postulated that after some distance, the activity of thrombin is 0. And that is why all the thrombin activity here is restricted to this site. And after the granule secretion in this site ends, we also see the disassembly of external region of thrombi. Is that clear?
Well, you mean that, well, I think that what is missing in my understanding or in your answer is that this is thrombin which initiates granule secretion, right?
Yes, yes.
Okay,
yes.
now we have all steps of this construction. Yes, thank you very much. So I don't know whether you still have time and force to continue with questions because in fact, I only started with my questions and I passed the word to our colleagues. So maybe I can ask some more questions.
Of course, I'm happy to answer. I'm happy to answer.
Yes. Then, well, sorry to return to my first question about the stochasticity and fluidity, and you explained how the stochasticity influences fluidity. But I remember also our previous discussions that the size of platelets you consider can also be different because rather often, and also what we did in our works, platelets are considered of the same size.
Yeah.
I think that you consider also platelets of different sizes with some distribution. And as far as I remember our previous discussions, you also told me that this also can influence the fluidity of the thrombus. Is it correct?
Yeah, that's a very interesting point. So unfortunately, for now, I cannot answer it completely. So you're completely right that in this particular model, all the platelets were equal, and in our later model with agonists, they are different, and in this study, which is already published, I think we had to show that in case if platelets are different in sizes, it doesn't influence our conclusions. Yeah, but we didn't do that. And I think it should be done because, of course, you're right. In case if we have distribution of platelet size, we can somehow have more fluidity, even in case if there is only just deterministic force. I think that's what you're trying to say.
Yes, this is my question. Yes.
Yeah. So unfortunately, I don't have an answer right now, but it should be done. Yes.
Yes. Okay. So another very interesting question for me, and of course not only for me, but you just stress the importance of this question, is how this core stops growing and then the shell is removed. And so I think that, and then you explain it with this limitation to thrombin inhibition, limitation to granule secretion. It's for me, it's completely different approach and a new one, because many years ago when we worked on related problems, our idea, our hypothesis, in fact, and also motivated by some experimental data, that in fact this is fibrin. You know, when there is in the thrombus core with this fibrin net forms, and in fact fibrin net it does not allow new platelets to attach. So at some point you see there is inside there is this fibrin net, then the shell is removed, and we stay with this core covered by fibrin, and this is why new platelets do not attach. This was the model, the idea, and then we realized it in the model, and we had this dynamics. So could you comment from your?
Of course.
It was already quite long ago. So, okay, our understanding can change. So could you comment on the possibility of this mechanism?
Of course, yeah, thank you so much. I was thinking about introducing this old result as one of the hypotheses that were suggested in the literature, because there are actually several hypotheses. And one is, for example, that the flux of thrombin is diminishing with time just because of coagulation. So there is this hypothesis is by Fogelson, I think. So your work, which is really wonderful work that I know, you're completely right that this might happen. Why we think that under the microvascular tree, why we think that this mechanism doesn't work? Let me try to explain how I understand it. So let's see the dynamics of this thrombus. So you see that so first it grows, then it becomes smaller, then we see some fluidity, and then all the rest of thrombus, so it just plastically deforms, and we don't see any cycles of the growth, right? And for example, in this video, which is pretty much the same, let's have a look. No, not in this one. Okay, in this one, we're now talking about microvessels. So what we see here is that, so here now it's big, right? We see some pieces detached, but then it generally becomes smaller and smaller. So what I'm trying to say is that in these types of dynamics, there is no like a single process that a big piece detaches and no growth occurs after that. Because in this hypothesis that there is a fibrin inside, actually here you can see where they detected fibrin in high amount. So this is the blue thing. So to my mind, in case if we have this small decrease of thrombus size, we cannot explain it in such a mechanism that that you discussed, that we detach some piece and we have some fibrin exposed and no platelets can attach. However, if we look at the bigger vessel, for example, in this one, the dynamics here looks more like what you're talking about. See, so here there is, it's not actually that good video, but here we indeed see that there is a huge detachments of some really huge parts. And in these bigger vessels, maybe sometimes the mechanism of the limitation is like you consider, but I'm not really sure, because even in this bigger vessel there is very often dynamics like this. There is growth, disruption, growth again, disruption, and like several cycles of growth, disruptions, and then it becomes smaller, and then it stabilizes. So that means that, to my mind, that means that it looks more like there is some fuel generated like from the inside, and this flux of the species, for example, ADP or thromboxane, with time it just diminishes. So it results in formation of this big part. Then it's disrupted by the flow, then it can grow again because there is still like secretion or synthesis or something like this. But then in time, it eventually stops just because the flux of the species declined. So this picture is now in my mind, but maybe for some circumstances, this hypothesis, as you suggest, also takes place. That's what I currently think about it.
Thank you very much for this interesting comment. I have one last question related to the last part of your work, where the vessel is completely cut. This is very interesting, and amazingly, we discussed a related question. We didn't do anything yet, but we discussed this question with completely also different approaches, more like continuous mechanics. The question I have here is that clearly whether the thrombus, the clot can stop completely, can occlude this opening. Clearly, it depends on the size of the vessel and on the flow velocity. So, do you have experimental data or some about how, in which vessels, in fact, the clot, this occlusion can really happen?
Yeah, that's a great question to my mind. Thanks. So first of all, what we know is that we observed these plugs, of course, in arterioles. So this was a lot of works on dogs, on people, even when we take biopsy. And for example, what I show you in the very beginning of my talk here is this picture. Famous picture, very beautiful picture. It's for the dog. So here is a small vessel. You can see single red blood cells here, and this is a platelet plug. So hemostatic platelet plug. You can see even some separate platelets here, as like dark spots.
So it's really for very small vessels.
So
right?
For very small vessels, it can 100% it can occur, and it should occur, and that is why I think most of all that is why hemostasis occurs, because they form these plugs. I know one paper, it's a recent paper of Michael and Alexandra Yakusheva. It's about high shear rates during hemostasis. And Alexandra made a very brutal experiment when she cut the semen vessel of male mice. And she saw formation of this plug, even though it is an artery. So it was a rather big vessel. And what is absolutely astonishing for me is that this plug formed just for 1 minute, pretty much the same as here for small vessels. To my mind, it's just completely mind-blowing. So it's so fast, just for one minute, huge artery. Of course, it's not huge like human arteries, but still, it's like several hundred microns, and it was occluded just for like one minute probably. You can find this data in her paper. I think it's very interesting.
Yeah, but when you consider, for example, this figure which you show here, or this other experiments here which you present, when this vessel is cut, is it completely opening? I mean, does it go on the free air, or it remains inside the body in such a way that there are some hydrodynamic resistance to the blood flow?
Great question. So in this case, there was just, I think it's like a cut with a scalpel, and after biopsy, they took these images. So in this case, you see that there was some opening, but the size of this area, which is opened, you see a lot of red blood cells here. It's hard to judge. In Alexandra's experiment, I think she cut this artery, which was exposed to the air. I think probably there was some water above or some buffer, but what I'm pretty sure is that she saw this vessel and she cut it with a scalpel, and it opened either to the air or most probably it opened to the buffer. So I don't really think that there was some tissues which prevented this loss.
Well, okay, thank you very much, Dmitry. It was a really brilliant presentation and very interesting discussion. I think that we don't have other questions from the audience. So thank you again for coming. Please keep us informed about your future progress. We are really interested to know that. Thanks also to our public, to our listeners. Have a good day, everybody, and goodbye.
Thank you so much. It was a great talk, and thank you for nice questions. We have to do the thing that you suggested. I'm really ashamed that we didn't do that. Thank you so much, Vitaly.
Yeah, thanks a lot. Bye.
Bye-bye.
Thank you very much.